Here we go. So we're looking at mitosis and cytokinesis. And so we're going to be looking at this division process here. And we only really have two big questions: is what is mitosis and what's cytokinesis? Like, how do they work? We know it at a ground level. We want to get a little bit more details though. So during that mitosis process, again, if you think about that big circle, it's just that little part here, that the cell's genetic material, it separates and it prepares to split into two cells. This is stuff we know already. They don't need to worry about that. Like, we should have that. The key thing here, though, is doing this accurately. If I were to give you, if you guys look on the back shelf there, if I were to give you every single book there, and I told you, I want you guys to write everything down there, word for word, with the correct spacing, you're going to make a mistake, right? Maybe you put a B instead of a D somewhere. It's going to happen. It might not happen a lot, but it's going to happen. So mistakes can happen. And when we are looking at your DNA, we are looking at millions of lines. So like, if you think about all those books there's like 100 books there. And I told you to replicate all that. You know, how many mistakes would you make? 1, 100, 10,000? For that, you making that mistake, it's not going to be a big deal. What happens if your DNA makes a mistake? Any guesses?
You end up having disabilities.
Potentially, could be a birth defect. Could be the cells doesn't work properly no longer. Absolutely. So when your DNA gets replicated wrong, bad things happen. Certain allergies, medical conditions can happen from a single line of your code being written improperly. And we'll look more at that later.
You ever watch Creation?
No.
It's with Paul Bettany. It's supposed to be who's that guy? Charles Darwin.
Oh, it's looking at the theory. Okay.
Evolution.
Yeah.
And he got with he was married to his first cousin and they had kids. And one of their kids got sick and they died.
Yeah.
And then you know how he's like a big furious, and he thought that like just because we're first cousins that you know their DNA kind of got then created a kid.
Could be, yeah. Yeah, well, I mean, yes, we know that's true, actually. But yeah, so the key thing here, and this is important, is mitosis needs to be done accurately because we know the consequences when it's not. So basically, what mitosis does, remember, this is when you have that one cell and it's getting ready to split into two. Here's my DNA material on each side. So it's getting ready. It hasn't split yet. It's still one cell that's just starting to rip apart. So with mitosis, we are increasing the number of cells in a multicellular organism as it grows. Again, most life is multicellular. But it all starts at one cell for humans. As well, mitosis can replace damaged cells. If you scrape your hand, your skin cells will undergo mitosis to fill it in.
It's on a page.
It would be in there. Yeah, I'm not fully certain where it is in there. So one thing to note though is, again, not every cell undergoes this cell cycle, right? Muscle cells, nerve cells, those don't divide. Red blood cells don't divide either. So when you get brain damage, like that's permanent, right? Like those cells aren't coming back. This is why if you have someone who's drowned for even a couple of minutes, you know, they drown, they stop breathing for 2 minutes, you resuscitate them, there's brain damage.
Come back and
I mean, it's a little more it's case by case, and like it's a little more complicated than that, but like if you get, potential brain damage, it could be it could be memory, it could just be like you don't have as much impulse control, for example. Okay, so let's actually start. So we know it's important to be done accurately. We'll look at how that is done accurately later, but starting out here, what is mitosis? Let's break it down. So it has a couple phases. The first is something called phosphase, and it's the biggest one. So we have a phase, we have a couple phases. The first one's phosphase or prophase, sorry. So basically, what happens is those cells chromatin, and if you remember from yesterday, the cell's chromatin is the relaxed DNA. So your genetic material, what it does is it clumps up. With all those novels there, that's a lot of information. What do? You put it into a book. So chromosomes are almost like the book, and these are the pages. So that chromatin turns into chromosomes. You've probably seen pictures of this in movies and stuff. We'll look at them actually later, but turns into chromosomes, DNA. So we have something called a sister chromatid. This is an identical copy of the DNA. It's a flash drive, right? I'm copying from one thing to the other. And I will show you a picture of this. It'll make more sense right after. Okay, so these images, these diagrams, you don't need to draw them. Those are given to you. But so with this profile, so what does this look like here? So again, those sister chromatids are structures that contain identical copies of DNA. So what those look like here, where's my picture? Is like this. So these are sister chromatids here. These are noticeably different than DNA. They are two separate entities. A really common misconception is because they kind of look similar, is that these are the same thing, and they are not. They both contain your genetic material, but they are not the same thing. These create the copy of your DNA. This is your actual DNA, if that makes sense, okay? This is my computer. These, the chromatids, are my flash drive that have the data.
So what chromatids go into, like, if you have a kid?
We'll end up going into that. Yeah, we'll talk about that throughout the course. So what ends up happening here is, so as this prophase continues, all these different things start happening. We get something called spindle fibers, centrioles, aster fiber. You don't need to know that. What you really need to know is we create something called a spindle apparatus. I like thinking about this as like a spider apparatus because it's kind of like a web. And it looks like a web. So like, most people eventually end up going spider apparatus instead of spindle apparatus. So it's just an easier way to remember it. So what ends up happening is this apparatus, and this apparatus is all of this stuff here, right? It's all of that combined. So that entire apparatus, that red region here, it starts to connect to those chromatids, to those copies of DNA. Yeah, this is metaphase is their second phase. So again, I will enlarge this photo here. I was just saying I'm going to enlarge the photo. Okay, so that's metaphase, that's our second phase. So again, they're lined up in the middle now, and they're going to start slowly pulling apart now. So leading into our final phase, or sorry, not our final phase, our second phase, which is anaphase. So this is when they actually pull apart. And if you're looking down here, you can see those two parts are now completely separated. So they're pulled from the middle and they work their way towards the edges. So they're no longer going in the middle. So they begin to shorten. And when it shortens, it brings it with it. So these chromatids separate and they move towards the poles of the cell. So anaphase is when the separation begins to go through the actual physical movement. Again, what's really starting to happen is these start pulling away, and that force is what separates these. Okay, so it's not that these are getting pushed there; they are being pulled towards the two respective poles here. And so finally, we are going to be at our last phase, and this is where it's really going to show the difference between mitosis and cytokinesis. Is if you look at the cell here, what's happened is it's just about to rip apart. It's at the most tension you can possibly get. Just a little bit more, and it'll split. So we are at the final phase here, which is telophase. So this is when those chromosomes have reached the poles. They've reached here, and now it starts to, they use the word decondense. You don't need to know that. It starts to split. Let's say what's actually happening here. It is starting to split. So we get two new nuclear membranes, and we get the nucleus starting to reappear. And that spindle apparatus, that combination of all those things, starts to disappear. It's done its job. It's moved the DNA over. It doesn't need to exist anymore. Okay, so again, walking through this whole thing here, so there's 4 phases, right? 4 phases: prophase, metaphase, anaphase, and telophase. So PMAT. Oops. And so this is just a enlarged picture of that. Again, you can see that it's just about to separate here. Again, PMAT. Here's our prophase. So this is when this apparatus starts to form and it attaches. This is menophase. This is when these line up in the middle. It's attached. It's going to get ready to start pulling. Anaphase, it has now started to pull, and these are now separated. And then finally, telophase, you can see it's just about to rip here, right? It's so close. You can see the tension. And then finally, what's going to happen is these are going to split into 2 cells. But this is not mitosis. This is cytokinesis. So cytokinesis is when it's fully split itself apart. Then those 2 cells can repeat the process, right? This is how you go from 1 to 2 to 4 to 8 to 16 to 32, and so it continues. It doubles, right? So the last thing here is just cytokinesis. So you have those 2 daughter nuclei, and they undergo the cytokinesis. Basically, in animal cells, those microfilaments, they tighten up so much that it just creates walls. It just tightens up, and now it's a barrier. Whereas in plant cells, it doesn't pinch itself. Instead, you grow a plate in between them. So that's the only difference between them. So cytokinesis isn't that deep. It's cytokinesis is just, yep, it's split. Okay, so I have a couple of questions that we'll go through, some exam style questions with this. That'll happen later though. Let's take a break from our notes and stuff. So what I want you guys to do here for the next syllabus, in fact, let me just write this down. Go. So what is it? We're 115 right now. So from 115 to, I don't know, like 135. Finish unit 3.2, the reading essentials. That's the stuff that's most of your notes. It's going to have your diagrams and the quick check. So there's the two things there. At 135-ish to, I don't know exactly how long, probably like 155 to however long it takes. We'll go over 3.2 questions. We'll have the break. And then after the break, we're going to try and get through 3.3. Maybe we won't. I don't know. We'll see. But that's just kind of what's happening. So for the next 20 minutes, not a lot of time, I know, is finish out that essentials and the quick check. There are two separate things there.
What's that? 130.
135, yeah. Okay, so just starting to go through some of this stuff. Here it is. So I got a couple questions here that we're going to go through. And there's a couple of them. Don't worry about number 3. That is printed out, given to you. That's in the essentials thing. I don't think you'll need a lot for question 2. You'll need a lot. You'll need like 6 lines for question 2. Otherwise, you should be good with like 2 lines each. So like, 2 lines between question 1 and 2. I don't know, like 6 lines between question 2 and 4. Then like 2 and 2 should be fine. So I will give you guys a minute for that. Okay, so question 1. So why does mitosis alone not produce those daughter cells? The reason is because mitosis only duplicates the nucleus materials. It only duplicates the DNA. In fact, you don't even need the rest of that. Just mitosis only does the nucleus material. The next one here is we have the 4 stages of oh, never mind, I didn't actually put it. So there's 4 stages here of mitosis. We have prophase, metaphase, anaphase, telophase. Again, PMAT is a good way to remember the order. Acronym it, right? It's a lot of information. You want to break it down. That's a nice way to be able to do it. PMAT. You can use that to make a story like I don't know, popsicles melt at temperatures or something. So prophase, that's when that nucleus membrane disintegrates, the chromosomes condense. Metaphase is when they are moved up to the middle, the equator, the middle. Anaphase is when they split. And telophase is when the chromosomes recondense and that the nucleus membrane and stuff forms.